Hello dear viewers, welcome to our channel Hahu class. In our last video, we have tried to see the general applications, definitions, the branches of microbiology and major breakthroughs or the findings that changed or that have revolutionized the study of microorganisms in the last 3 centuries. In our today's video, we will be just starting to demystify viruses. Okay, viruses. In virology, or we will be just focusing on virology. Virology is one of the sub-branches of microbiology. Okay, and just similar to microbiology, general microbiology, in viruses, until the 20th century, in the late 20th century, the study of virology was not known. It is a very old disease on one hand. There are diseases that happened to be there before 2,000, 3,000 years ago. But only in the 20th century that knowledge about viruses started to emerge. So virology is one of the basic sub-branches in microbiology. Okay? In our today's video, we will be just looking at what virology is and the landmarks, very important landmarks that are related with virology, particularly those major findings that were that have changed the world when we talk of viruses, the virus world. So 20th century is the most important landmark when it comes to viruses. So we'll try to define viruses, we will try to see the landmarks in virology, and we will see what are the properties of viruses. Okay? There are a lot of properties or the characteristics that make these entities very particular related to other infectious agents like the fungal, protozoal, or bacterial agents. So we will see in detail the properties. With the early development of virology or study of viral diseases is historically is a very new science. Essentially, it is very old. It is very old because there are diseases that are known to be there in humans, in animals, in plants, and whatever. Infectious agents are available throughout human history. So it is very common to see diseases like polio, like rabies, and many other viral diseases during 2,500 years ago, like that, before Christ. Okay? So the diseases, their infectious process, and their symptoms, and even the treatments, even vaccinations have been known. They were known prior to the discovery of infectious agents themselves. The diseases were available long ago, but the infectious agents were not known. So only the discovery of the microscope helped. Only the discovery of microscope helped figure out what kinds of infectious agents are available around the world. Well, that's about general microbiology. In terms of virology, this is a very new one. Only in the late 19th century, this disease infectious agents were known, started to be figured out. Okay, there are some evidences that great epidemics happened so long ago. This is AD 165 to 180 AD, and plus 251 to 266 AD after days of Jesus Christ, which severely weakened the Roman Empire and aided its decline, which may have been caused by measles, diseases such as measles and smallpox in human beings. Okay, so the diseases were available, generally speaking. If you see through, look over here, okay? The first progress in preventing viral disease came years before the discovery of the virus. That's what I was saying, okay? During the 17th century, Edward Jenner, who tried to discover the cowpox, was surviving or working during the 17th century. So, Edward Jenner has published about 23 successful results of vaccination in 1798, this 18th century. He didn't understand the nature of smallpox, for instance, at that time. He didn't know that this disease was, the disease which is called smallpox was available around, but he was sure there is some infectious agent that caused the great epidemic of smallpox. Deaths of so many people, okay? Look here over here, for instance. If you see this picture, poliomyelitis through the age has been known. For instance, before 1200 before Christ, it was there. But the discovery of this poliomyelitis virus was only there at around 1968. This is what I wanted to say. So, viruses in the human history, for instance, rabies, polio before 1,500 years of before Christ, smallpox, as I said, also before Christ, and some emerging viruses still there. So they were long ago there, and still they are continue to be there. So this is you can see the development of virology throughout the years. This is the, as you see, the golden age of microbiology started only in second half of the 19th century. In the second half of 19th century, when some very important people like Louis Pasteur, the father of modern microbiology, okay, had started, and one knowledge builds upon the other. The other knowledge builds upon the other. The other knowledge builds upon the other. And now this is the era of molecular biology today. The era of molecular biology, or the era of more advanced studies related to what? Related to virology. Especially the last 20 years. The last 20 years are very fundamentally breakthrough. They are fundamentally breakthrough in issues related to what? Related to more advanced studies related to microbiology in general and virology in particular. So, in virology, very important developments were made in the late 19th century. The first person who embarked on virology was Dmitry Ivanovsky. This is a Russian scientist who reported in 1892, around 1892, that it was possible to transmit tobacco mosaic disease from diseased to healthy plants using filtered leaf extract as inoculants. Okay, in this experiment, he demonstrated that the filters which were used by Ivanovsky were characteristics of viruses which can infect animals. This was in 1892. The next very important timeline in virology is a time 1898 by Martinus Benjanrik, who demonstrated the filterability of agents of tobacco mosaic disease. So he was the first person, okay? He was the first person to recognize the virus are reproducing the entities that are different from other organisms. So when he undertook this experiment, he was unaware of the work of the Russian scientist Ivanovsky. So he demonstrated that the disease could not be due to toxins, as the filtered sap from infected plants could be used for serial transmission of disease without loss of potency. Look at this. This is tobacco mosaic disease. Okay, this tobacco mosaic disease is the upper picture. The lower is tobacco. The next very important landmark in microbiology, or specifically virology, is in 1898, when two scientists, Loeffler and Frost, identified the first filterable agent from animals, the virus of foot and mouth disease, or FMD. So this time was a very important finding in the same year. It's the same year. The next important landmark is 1851 to 1982. There was a person named by the name Walter Reed. He tried to see that during the Spanish-American War, subsequent building of the Panama Canal, American deaths due to yellow fever were very high. Okay, the disease also appeared to be spreading slowly northward into the United States. So through experimental transmission to mice, in 1900, Walter Reed demonstrated that yellow fever was caused by a virus spread by mosquitoes. So 1901 was a time where publication was made for the first time in that situations. The other very important time is 1908, where Ehrlichman and Bank, two individuals, first discovered a virus producing leukemia in chicken, or chicken leukemia. And in this, the scientists demonstrated the oncogenic potential of a filterable agent, which is avian leukosis. Origin of virus. Where the viruses come from? What are the theories that are synthesized or that are postulated to discover what the viruses are? Where do they have evolved from? Viruses have evolved to a stage where they are considered to be among the most efficient and economic forms of microbial life. The most efficient, the most economic forms of microbial life. And three types actually. DNA viruses are there, RNA viruses are there, and there are certain viruses that utilize the combination of DNA fragments and RNA fragments at times. Most often, DNA viruses and RNA viruses exclusively take the vast majority of composition of viral diseases and the viruses themselves. However, some viruses can utilize both DNA and RNA during replication. Okay? We will see when we study about replication. The differences between these groups are very significant and may be indicative of independent origin of each group. So what are three postulates or the theories that are known as of today? Are the first thinking is, there are three theories as I have said. The first thinking or postulate is that the viruses might have originated and evolved in parallel with primordial life forms. In the evolutionary process of living things, viruses were there. as early as so many millions of years of age. This is the first. So, in parallel to bacteria, in parallel to fungal, in parallel to vertebrates, in parallel to invertebrates, viruses have all along have been there, surviving. They were surviving from day one. From the day one, parallelly as it is in other forms of living organisms. This is the first theory or the first idea that happened ever. The second thinking or theory that is hypothesized is viruses might have arisen from segments of cellular nucleic acid of a cell. You know it, okay? Multicellular organisms have nucleic acid within the cell. DNA fragments and RNA fragments which are packed in the nuclear membrane are in there. Then, what it does say is that they might have arisen from segments of cellular nucleic acid which are acquired the ability to replicate at the expense of host cell. At the expense of host cells, the fragments might have evolved from the cellular organisms or the cellular bodies themselves, and by so doing, those cellular fragments spontaneously evolved to what we call the viruses. This is the second idea. Third. The third point is they might have risen from living organisms which are gradually losing their genetic information until they became totally dependent on biosynthesis pathways of their cells. What does it mean? There are other free living organisms. From free living organisms, gradually, those free living organisms, they may lose some genetic information. Okay? Some genetic information were lost, some mutations happened which deviate from the original DNA organization. Then that fragments that detach from the original fragments of the DNA or cellular subcomponents might have risen to what we call viruses. So this three theory is in place. Next point is properties. You know the properties of bacteria. What are particular issues that make these viruses different from bacteria or other living organisms like multicellular organisms like fungus and prions? So, in this one, they are infectious agents, mind you, they are infectious agents, or sometimes called they are parasites. You know parasite. They are parasites which consist of nucleic acids, surrounded by one or more protein shells. At times, those protein shells themselves, which are known as the capsid, are sometimes surrounded by other cellular components, which are called the envelope. Actually, the envelope might be there in some viral types, and the envelope might not be there in some other types of viruses. But generally speaking, we can say that nucleic acids, which are either DNA or DNA surrounded by the protein shell, and sometimes having another additional type of coverage, which is the envelope, and in some other viruses which have no envelope. This is the definition of viruses. So generally speaking, they are, number one, they are intracellular, mind you. Viruses are intracellular. There are obligate intracellular parasites. Obligate intracellular parasites. Which means that obligate means what? Without the presence of another cell which to depend on the viruses can never replicate and multiply themselves. They are intracellular, they need another cell to depend on, and that is obligate, that's a must. That's a must, okay? And they can replicate only within the cells. There is a nucleic acid do not encode many enzymes, very few enzymes. In certain viruses, only 2 or 3 enzymes are enough to do all the job. And in other viruses like the pox virus, like hepatadenoviruses or hepatitis B virus, you will need tens and even hundreds of enzymes and factors. And other viruses like HIV virus, like COVID-19, for example, SARS-CoV-2, SARS coronavirus 2, and many other viruses, very few enzymes, 3, 4, sometimes 5, 6. Structural and non-structural proteins may be there. And in other viruses, as I have said, in complex viruses like smallpox, like pox virus, or like hepatadenoviruses, you will need 100 and many more enzymes to be there to do their job. But the issue is, in all of them, it is the same. They are never independent by themselves. They depend on cellular machineries, and the cellular machineries will be utilized by the virus. So redirection of the host cell cellular machineries, and at the expense of those cellular machineries, the viruses redirect. They redirect the different coenzymes, different factors for themselves, and they use to produce their own the viral products, viral product, gene product or viral product. Well, it continues. As we said, they have infectious cycle. Infectious cycle means recognition is the first one. We will see later. The viruses must have receptors and co-receptors, and those receptors and the co-receptors must be, it is compulsory to have those receptors and the co-receptors, and those receptors and the co-receptors must be recognized by the cells being invaded, followed by attachment of the virus receptor and the co-receptor onto the surface of the cell, and followed by penetration of the cell, the virus, I mean, penetration of the virus into the host cell, then uncoating. We'll see later during the replication. They uncoat, they do replicate. After replication, there is assembly. After assembly, there is release. Okay? So, this is what we call the infectious cycle. So, all of the viruses go through this stage: recognition, attachment, penetration, uncoating, replication, assembly, release. This is the step. The other is they are transmissible. They can transmit horizontally and vertically. Horizontally means from one person to another person of the same species, or from animal to another animal, or from one plant to another plant, horizontal. Vertical is from mother to child. Okay? From mother to child, they are transmissible. The other is, I have already talked about this issue, they do redirect genetic and metabolism apparatus of the host cell for the use of, for utilization by the virus. They have almost nothing. They do enter there. They have some starting materials to begin with, some starting materials, and those starting materials will be used as some raw material, but most of the other enzymes, essential, they are missed from the virus. However, they can utilize, they steal from the host cell okay, by integrating into those cell DNA, okay, the DNA, a specific segment of the DNA, and thereby they extrapolate, okay, they extract the machineries, enzymes, factors, cofactors, or whatever that are very crucial for the replication of the virus themselves. Then they do their replication and they go out. They go out and the cycle continues. So they have major features of cellular organisms. For instance, they have life cycle, as I have said. Okay, recognition, attachment, entry, uncoating, replication, assembly, and release, like that. That's what we call life cycle. This makes what? This makes the viruses to fulfill the criteria of a living organism. Okay? In any living organism, there is a cycle, be it vertebrate, invertebrate, or whatever. So this is it. The other is they have a defined stage of development. A defined stage of development during their stage. Okay? Replication stage, early replication, late replication, okay? Assembly stage. Different stages have particular characteristics that is associated with that level of development. The other is organization. In any living organism, there is cellular organization, okay? There is a cell, tissue in multicellular organisms, as you know. There is a cell, a group of cells becomes what? Tissue. Group of tissue becomes what? Organs. Then organ systems, and then organism, similar to that, okay? And if you see the cellular components and subcellular components, you can see that there is a cell. Within the cell there are a lot of cytosol organelles within, and then the replication starts from there, and the growth starts at the second stage. Similar to this one, they have a particular scenario where they can fulfill the development of a living organism by what? By reorganization and organization. Okay, a particular type of organization which depends on a specific type. And the other is genetic variation. We will see genetic variation later in our discussions. However, they do not possess some features. One, no machinery of metabolism. For a metabolic process, they are incomplete, half. Okay? They have some components, very few, ingredients, but missing other very important ingredients. And those missing ingredients will be fulfilled from where? From the cell. That makes the virus to put very difficult into a living organism and a non-living organism. Outside the cell, they are simply particles, okay? Okay? For example, if you release a virus, your common cold, or these days COVID-19, another person around may catch that virus. But if the person is not around, what happens? The virus will stay some 3-4 days, depending. Some viruses can stay for months. Other viruses have a very short lifespan, especially if exposed to UV radiation like sunlight, if exposed to high temperature, if exposed to desiccation. What happens? They have a very short-lived lifespan. Others have long-lived lifespan. Despite this difference, the viruses are essentially the same. Outside the cell, they become almost non-existent, simply crystals or any material, as if they are, non-functional. But by the time they get the appropriate host, susceptible host, by the way, susceptible host means that is not immunized. Unless that happens, the virus becomes very silent, very inactive for a few days before it dies off. It can die, but the days depends on. It depends on the type of the virus. In terms, some viruses, they are very short-lived, 1 day, 2 days, 3 days, or even hours. Okay, especially if exposed to desiccation and sunlight. This sunlight is dangerous. for many viruses. Otherwise, other viruses, they can withstand even very harsh environments like the deserts. For how many days? For some specified period of time. After that, they die. They die out. This is actually true for bacteria as well, true for fungal infections, in many infectious agents, okay? So this is it. What makes viruses different from toxin? There is what's called toxin. Toxin and antitoxins, antidote, for example, bacterial toxins, you remember? Okay, Shiga toxin, okay, botulinum toxin, tetanus toxin. So those toxins have some difference from the virus. In the case of toxins, despite their similarity to cause disease, toxins, they can effectively bring about diseases in susceptible hosts. But the issue is, in the case of toxins, mind you, they never multiply. They never multiply. Okay? If, for instance, the LD50 of a toxin is, for example, if it is 1000, the amount of, or the dose of a given infectious agent that is capable of bringing deaths to 50% of the population is called LD50. So, if the LD50 of a given infectious agent is 1000, then that specific amount will be inhaled or taken by a vulnerable host or susceptible host, thereby bringing about disease. But the difference in the case of virus and this one is, in the case of the toxin, that specific amount of toxins that can bring about either mortality or morbidity can never multiply within the host cell, never multiply. The amount that is injected will still be there, but that amount can bring about mortality or morbidity, it depends. But in the case of viruses, however, one virus may enter into your cell, it will infect one cell, it enters, multiplies, it does assemble. By the time it leaves the cellular surface, it will directly go to another cell, another susceptible cell. So another susceptible cell will take up to the virus, and the cycle continues. Once the virus will give to 2 to 4, 4 to 8 exponential, and by the time it reaches a threshold, the cellular component will either explode. What about plasmid? You know plasmid in bacteria. Plasmids are extracellular genetic material that code for different activities. For example, resistance to antibiotic, remember, okay, for sex plasmids, genetic materials, they may be there. So those plasmids, as we know, they are very small fragments of DNA of a bacteria which happen to be outside the normal genetic material of the bacteria, but they have some activity. They have some activity, they have some mission to undertake or to execute. So the difference between plasmid and a virus is, in the case of plasmids, they do self-replicate, okay? They do self-replicate, but they lack protective structures. Unlike the viruses, they lack protective structures, but they can replicate in massive amount, either intracellular or extracellularly within the in vitro environment. The other is, they differ from other intracellular parasites like bacteria. You remember, for instance, tuberculosis, Mycobacterium tuberculosis, Brucella are essentially intracellular. They are essentially very intracellular. Viruses are obligate intracellular. So both of them are intracellular. What's the difference then? In this one, okay, infection is maintained by cell. The infection is maintained by cell, and it does not have eclipse period. The other property of a virus is every species of life, be it a bacteria, be it a human being, be it an animal, be it plants, all of them, they are always vulnerable to at least some sorts of viruses. They are vulnerable to certain types of viruses. So every species on Earth from A to Z, they carry viruses. They carry viruses. The other property of a virus is many viruses are harmful. Many viruses are harmful. Very few actually. Very few are symbiotic. In bacteria, the vast majority of bacteria are what? If you remember, they are symbiotic. At least they are not harmful. Very few are pathogenic. Okay? In the case of the virus, there are around 4,500 different types of viruses. And this gives advantage to the host. For instance, drug resistance, gene transfer, virulence factor, for example, as carriers of some genes. Many viruses are utilized especially genetic recombination. Especially in genetic recombination or genetic engineering, many viruses are utilized for the sake of vaccine development, for example. For the sake of modern vaccine development, some viruses are used as carriers of a certain genetic material from another organism, be it a bacteria or a virus, and they will be used as what? As a carriers of a certain genetic material to be utilized for some activity like genetic engineering or vaccination development. So the other thing is the genomes of the viruses which infect animals are smaller than those of prokaryotic cells. It ranges from 2 kilobase pair, 2 kb or 2 kilobase pair, and some of them can go as much as 200 kilobase pairs, even 400. Some viruses like pox virus, it carries around 350 like that. And in the case of adenovirus, it carries about around 350 kilobase pair. This pairing can go up to 3 billion pair in human being, and up to 35, sometimes around 30,000 genes distributed over 23 pairs of chromosomes in human beings. In the case of the virus, very small, you can see. The maximum is around, maximum 400 kilobase pair, mind you, okay? But the smallest viruses are also available, okay, like picorna virus. They are very small in size. Many viruses are on the middle, we will see. Most of the viruses are on the middle between around 80, 100, 200, 60, around, they move around that. But a few viruses like picorna viruses, they are very small in size. But very few also are very large in size. But by the largest, we mean very few ones, like pox virus, like adenovirus. Okay? So, in terms of size, this is what I can say. The other property is, in most of the viruses, the nucleic acid is present as a single molecule. either linear, most of them are linear, some of them are circular, and in some RNA viruses, nucleic acid occurs as a segment, as a segmentation. For instance, in paramyxovirus, viruses, avian influenza, which you call avian influenza, there is segmentation around 8 segments of the viral genetic material, or the nucleic acid is there, about 8 segments segmented. And also in Rift Valley fever virus, okay, in Rift Valley fever, also segmentation is there. That segmentation is, we will see, is very important, okay? There is what we call genetic shift and the genetic drift. So for mutation and the genetic shift and the genetic drift, that segmentation is very important. But the vast majority of the viruses are unsegmented. And many are linear. So many are linear, there are a few circular. The other point is, they have envelopes. Besides the essential components of the viral components, which are the nucleic acid and the capsid, the envelope may be there. That envelope is sometimes partially synthesized by the viral genome itself, from the viral genome source, and sometimes they utilized from sources of the host machinery, but the envelope is there in a few viruses, some viruses, particularly in RNA virus, okay? This is true in RNA, many RNA virus. Many RNA virus, they have envelope, especially circular viruses, okay? Circular virus. The other characteristic is the viruses cannot replicate on inert media. In bacteria, you can grow any material that contains some essential components as a food. They can grow easily. But in the case of viruses, you cannot grow in inert media. So you need a viable host cell, what you call this, we call cell culture. Cell culture that is produced in immunology laboratory. You will produce the cell culture. With that cell culture, you will inject the viruses, and the viruses enter into the cell and they replicate. Besides cell culture, some viruses can grow in animals, in live animals. This is what we call animal inoculation. Animal inoculation. In rats, in guinea pigs, you will inject, you reproduce the virus, and you will harvest the virus to be used for what? To be used for either vaccination development or for other activities. So viruses never grow in inert media, unlike bacteria and fungus. They need a viable cell lines, primary, secondary cell lines that are used for in vitro viral cultivation in virology laboratory. Okay? That said about the property of virus, let us compare and contrast the differences between a virus and differences between a bacteria. In terms of size, the largest virus is almost comparably equal to the smallest bacteria. Mycoplasma is the smallest known bacteria, like Mycoplasma mycoides subspecies, mycoides large family, Mycoplasma mycoides subspecies, mycoides small family, you know this, CCPP and CBPP, okay? and also mycoplasma disease, which is a pneumonia, a disease of pneumonia in human beings. Those types of bacteria are by far the smallest in size. They are around 0.3 micron, 0.3 micron. That is the smallest bacterial size. When you come to the virus, this size is the largest of the class. In the largest virus, you will only be able to find a virus which is as large as 0.3 micron. Actually, no virus is measured in microns. All of viruses are measured in nanometer, okay? Mind you. Always we measure, when we describe about bacteria, we use micron, you remember, right? In the case of virus, we always, almost constantly, we use nanometer. The largest bacteria, the virus, I mean, is almost equal, comparably equal to the smallest bacteria, mycoplasma. Number two, let us see growth. In the case of growth, okay, they can grow on non-living organisms. You can produce bacteria, you can multiply bacteria in nutrient agar, on blood agar, makonki agar, or specific agars, selective agars, many types of agars. Nutrient brose even. Nutrient brose also. Okay? You can grow. All of these media are what? They are non-living media. Inert media. They are. However, the virus cannot grow on inert media unless you do either primary or secondary cell lines in virology laboratory. Well, that's called cell culture. We talked that. The third point, nucleic acid. The case of bacteria, almost all of bacteria, they have both DNA and RNA. But the DNA and the RNA are simply floating within the cytosol of the bacteria. Okay, they're simply floating. But in the case of multicellular organisms like yeast, like vertebrates, like humans, mammals, what happens is the nucleic acids, both DNA and RNA, are packed into double-layered nuclear membrane. You know this. So that nuclear membrane is a protective of the delicate materials, which are the DNA and RNA. In the case of the bacteria, they are simply floating over there, okay, here and there. Actually, all of the other organisms are the same in the case of bacteria. But in the case of the virus, either DNA or RNA, never both. Number four, in the case of nucleic acid, the nucleic acid of bacteria is non-infectious. That means you need the central dogma of nucleic acid, replication of nucleic acid. In DNA, nucleic acid, there is transcription. Okay? From DNA to messenger RNA. And from messenger RNA, you have translation to proteins, right? In the case of some viruses, especially single-stranded RNA virus, particularly single-stranded RNA virus, what happens is, if you give the messenger RNA to some vulnerable or susceptible host, that vulnerable host will directly take up and directly multiply, the directly production of the relevant structural and non-structural products. or protein materials. Simply, the messenger RNA is directly infectious. We will see the details later when we study about replication of viruses. The fifth comparative analysis is ribosomes. You may expect ribosomes are available in the case of bacteria as free living, okay? Free living without having rough endoplasmic or smooth endoplasmic reticulum. In the case of multicellular organisms, you know that there is rough endoplasmic reticulum where the ribosomes attach, and a smooth endoplasmic reticulum or AR which doesn't attach. But in the case of bacteria, no. They simply float around, as it happens in the case of nucleic acids. So the ribosomes are absent in the case of virus, but present for bacteria. The sixth comparative point is metabolism. Okay? True that bacteria always do metabolism by themselves. But in the case of virus, metabolism never happens. You know metabolism, okay? The other point is sensitivity to antibiotics. This you can expect. Many bacteria, 99% of bacteria, are sensitive to appropriate type of antibiotics that is designed for that group of bacteria. They are vulnerable to that. In the case of virus, however, that doesn't happen. No any type of antibiotic can help grow, can help kill, or at least stop. So in summary, we have tried to see the landmarks of virology, 20th century, is very important time as so long as virology is concerned. Particularly the last 20-30 years is a very, very important timeline when you consider the more advanced studies related with viruses, okay? So we have seen the detailed issues that are characterizing viruses when compared to other infectious agents. So this is the video. This is the video that is related with the introductory concepts in virology. For today, we will discuss other subsequent issues in the next video. Thank you very much for your attendance. Thank you.